Moving on from talking about DNA replication, transcription, and then translation, we need to move into the cell cycle, because with all that DNA, it encodes what needs to happen. The cell cycle allows the cells to reproduce to make more cells, replicating that DNA. So we'll start with this cell cycle. Cell cycle has a couple different parts. You can see the M here. That M is where mitosis is actually occurring. You then have G1. That's a growth phase. During G1, the cell is growing after it's gone through mitosis, making sure the organelles are the full size, make sure everything is working properly. Now, if that cell is going to reproduce again, so basically go through mitosis again, it'll enter into the S phase. The S phase, or synthesis phase, is when the DNA is replicated. You'll start with the normal double strand, and then you'll make a copy of that. So you have two exact copies of that double strand, which means this cell, for a short period of time, will have double the amount of DNA that should be there. But then once the S phase passes, you get into the G2 phase. This is a growth 2 phase. This is when the cell starts to prepare to go into mitosis, starts to increase the amount of materials and organelles and get everything set and ready to divide into two separate cells. Now, if the cell is not going to grow and go through mitosis again, it enters into this G0 phase or G0 phase. In G0, it's just going to kind of grow to hold and then wait. It will not get into the actual phase of S phase and then G2. Now, in growth phases and synthesis phases, there's not a whole lot to talk about from our perspective. What we need to focus on from this phase is going to be mitosis, the M phase. So the G0, G1, S, and G2, not a lot's going on from what we need to worry about. Lots happening in the cell. We want to focus on the mitosis phase. Mitosis will undergo 4 main steps. We have prophase, metaphase, anaphase, and telophase. If we start with prophase, in the prophase step, we're going to notice that the actual nucleus has the nucleoli disappearing. So you can see the nucleus here doesn't have a nucleoli. Instead, the chromosomes are thickening. They're getting ready. The centrioles are here and here. Those are moving towards the opposite ends. Now, if you notice, if you go to this pro-metaphase, which is just before metaphase, those centrioles are on both edges. They're ready to create the network that pulls the cell into two. Also, what's going on is that mitotic spindle is forming. That's these lines. These are the mitotic spindle lines. It's creating almost like railroad tracks to allow the different organelles and parts to separate and the chromosomes to separate out. It helps give a pathway for the chromosomes to follow to each one of the new cells. So prophase, kind of getting things ready, just about ready to line up, but we're not lined up yet. Metaphase is where the lining up occurs. Metaphase is distinct because you'll have the chromosomes lined up right down the middle of the cell. They're lined up in the middle, getting ready to split. So the chromosomes are clustered right in the middle. They're getting there and forming what's called the metaphase plate. So it'll be that line right in the middle of the cell. Now, these are the two chromatids still joined together. So they're joined together by the centromere. They haven't separated out yet. So you have them still joined in the metaphase. Next after metaphase is going to be anaphase. Now in anaphase, you can notice the actual chromatids have pulled apart. Those things that look like the X, the chromosomes, they've split apart. The sister chromatids have pulled one to each side. This allows for the starting of the separation that will eventually become two completely new cells. But remember, mitosis makes exact copies. That's why the sister chromatids pull pull apart. They're exactly the same. Both cells will be the same. And this is also when cell kinesis just starts to appear. There's not much happening yet in that, just starting there. Now, as we keep going, those chromatids keep pulling apart. You can see down here I have, say, motor proteins. Motor proteins are pulling the chromosomes. Basically, you've got a little tiny engine pulling the chromatid along the cytoskeleton. Telophase is the last phase. In telophase, we now have two nuclei starting to appear. We haven't fully made the cells yet, but those chromatids are starting to unravel and create the actual nucleus of a new cell. That new nucleus is being formed by a new nuclear envelope and a new nuclear membrane. You're having sections of rough endoplasmic reticulum form around the outsides as well. You can see here and here our new rough endoplasmic reticulum starting to form outside the nuclear membrane. So getting everything ready to go. Centrioles are replicating this time. Each cell needs two centrioles. Well, the cell had two, they split apart. These one on the ends now are replicating to have two new ones. So that way, once you go through cytokinesis, which is the pinching of the cell membrane here, that pinching will continue until it goes all the way through, creating two completely new cells. By that point, we'll have our new pairs of centrioles. We'll have our nucleus formed with the full set of chromosomes. We're going to have our rough ER formed. We're going to have the other organelles in there. We're basically creating two exactly duplicate cells. By the time we're done here with telophase and cytokinesis, two cells are ready to enter growth 1, growing after mitosis. They're exactly the same, perfect duplicates. And this is how most of the cells in your body will go through making new cells. Mitosis after mitosis, all these mitotic processes are occurring. But there's a few cells that won't use mitosis, mainly the gametes, egg or sperm. So mitosis summary here. What do we accomplish? Went from prophase to metaphase to anaphase to telophase, exact duplication. We start with 46 chromosomes, 23 pairs. Then, as it's getting ready, you duplicate to 92 chromosomes. Well, it makes sense. 46 times 2 is 92. Enough chromosomes for the two completely finished daughter cells. Divide into 2, back down to 46. So yes, momentarily you have extra DNA inside that one cell because it replicated the DNA, made a copy. But it didn't divide quite yet. Once it divides, we're back down to 46. So the normal cells typically have 46 chromosomes, 23 pairs. But during that duplication, when getting ready, it goes up to 92, goes back down. But again, exact copies. Except in meiosis now, not exact copies. Meiosis is used by the sperm and egg generation in the bodies. It still has a kind of similar feel to it. As you notice here, we have prophase 1, anaphase 1. We have telophase 1, we have metaphase 1. Well, I keep saying 1 because it has to happen twice. But one of the big differences is looking at the actual chromosomes. Notice they mix up the DNA. The one that's here in blue has a little red tip to it. The one in red has a little blue tip. In meiosis, you'll have swapping of genetic material between the chromatids. This means there's going to be genetic variation. Not everything's exactly a duplicate. Things get mixed up a little bit. We're not talking massive amounts of swapping, not whole entire things, but pieces here and there get mixed, get swapped, get changed. So we still go through the same steps from prophase to metaphase to anaphase, telophase through cytokinesis. That 1 became 2. Now, if you notice though, we'll continue from this first process into the second process. In meiosis 2 now, we have prophase 2, metaphase 2, anaphase 2, telophase 2. It's a second series through the same exact process. Meiosis as a whole basically goes through these twice. by the end, the actual developing cells at the end, now we're simply looking at a new cell with half the amount of chromosomes. Because if you notice, we don't have the same full amounts. So from here, one cell started, divided 2. Those 2 then went along the whole process again, divided again to make 4. In meiosis, one starting cell becomes 4 finished cells at this point. So in the case of sperm production, one becomes these 4 sperm. In the case of an egg production, so we're on the female side now, one cell becomes 4, but only one's a functional egg. 3 are called polar bodies, they don't function. So it's still 1 to 4 for the genetic material though, which means you still duplicate, but then you cut in half and cut in half a second time. So we end up with half the chromosomes through meiosis plus some genetic variation. The whole idea, making sure that when the egg and sperm do combine, it goes back to the number 46 properly. So by taking half and half, you get a normal cell again. So the idea of mitosis and meiosis. Mitosis is used for the majority of all the cells. Exact duplication, 1 makes 2. Meiosis, you're finding the gametes. 1 makes 4, not an exact copy, and half the chromosomes.